The effect of various oils on adaptive mutations in Saccharomyces cerevisiae and E. coli as cancer models. In 2006, cancer was the second leading cause of death in the United States behind heart disease. In 2003, cancer was the cause of 23.3% of deaths in New York.
Cancer is the result of a long process that begins when one of the cells in an organ or tissue becomes damaged or altered in a way that causes it to break free from the normal controls that allow our cells to work together in harmony.
Most cancer results from somatic mutations, which are DNA mutations that have not been inherited and occur at some point after conception in body cells that accumulate over a person's lifetime. Adaptive mutations, also called directed or selection-induced mutations, are spontaneous, that occur during periods of prolonged stress as specific responses to environmental challenges. The key feature that distinguishes adaptive from random mutations is the specificity. They appear to only occur in the genes that are under intense selection. In multicellular organisms, adaptive mutations contribute to a breakdown of somatic cells from negative growth regulation and have the potential to lead to cancer. Most adaptive mutation information is gained from Saccharomyces cerevisiae and E. coli as model organisms. Many researchers suggest that various natural plant extracts have properties that can stop adaptive mutations and kill cancer cells. These oils include basil, cinnamon, clove, frankincense, lavender, oregano, tea tree, rosemary, and thyme. Reddy in 1997 tested pryl alcohol and limonene derived from lavender and orange to determine their chemosynthetic properties against liver and/or lung cancer. The study was designed to test the efficiency of 40 and 80% tolerated dose levels. It was found that alcohol greatly inhibited tumors in animals. Hall in 1995 tested the findings of recent studies that suggest that adaptive mutations in non-dividing microorganisms can help understand the mutation origins in tumors. The graph shows the observed and expected number of colonies in the strains of E. coli. If the colonies observed in the experiment were random, they would have stayed consistent throughout the study. The results showed the number of lactolase mutants increases with time, suggesting the mutants were adaptive. Many researchers suggest that oils of natural plant extracts have strong antibacterial activity when tested against the bacterium E. coli. Friedman in 2002 tested the bacterial activity of 96 plant oils against E. coli using an improved method of microplate assay. The most active oil against E. coli was oregano and clove oil. The graph shows the time-dependent accumulation of adaptive mutations. At start time, no pre-existing colonies were on record. If a deletion of a certain gene results in a frequency of adaptive mutations, as seen in gene 1, this indicates the gene is acting in an antagonistic pathway. If the adaptive mutations is decreased, it can be inferred that the gene positively influenced the adaptive mutation. In this study, two main experimental groups, E. coli and Saccharomyces cerevisiae, will have 8 variable groups and the control with 10 trials each. The experimental setup for a study of adaptive mutations has to meet 2 main criteria. First, the populations of cells has to be kept in a growth-limiting environment with the application of non-lethal stress conditions. Secondly, the growth-limiting stress application should represent a strong pressure favoring the adaptation. The colony assay is used to plate adaptive mutations. Cells are spread using a nutrient-lacking compound. This result is a rough monolayer of cells that, when recultured, will exhibit uniform conditions. Lactose peptone and copper sulfate are both environmental conditions that result in adaptive mutations of E. coli and Saccharomyces cerevisiae, respectively. For both E. coli and Saccharomyces cerevisiae, each culture will be spread on their designated agar and incubated for 48 hours. Then, using the colony assay method, the mutant colonies will be spread on agar with 1 paper filter disc of oil each. The zone of inhibition will be measured in millimeters using a ruler after 48 hours of growth. The budget for this project includes the E. coli strains, nutrient agar, Saccharomyces cerevisiae strains, copper bromine, copper sulfate, lactose peptone agar, and the 9 plant oils. The goal of this study is to determine if various natural plant extracts are effective against adaptive mutations in E. coli and Saccharomyces cerevisiae.